So we're moving on in our second section to a discussion of the nervous system. This will take a few weeks. It'll cover a few sections, and the point of these slides is to learn about the branches of the nervous system. Neurons are organized in a specific way to achieve sensing and motor goals within the body. We'll talk about the various cell types that are found in nervous tissue, the formation and appearance of neurons. You will see a neuron in lab this week, in Lab 1. We'll start to look at the brain and the gross anatomy of the brain, how the brain is organized into structures and substructures, the surface features of the brain, the main divisions of the brain. We'll talk at length and in less detail than I would like about that before moving on to discuss the eye. The functional organization largely follows the form of the brain. This is less clear with nerves because form and function aren't obviously linked with nerves. In a lot of other places they are very tightly linked. And that's wrong. That's next section. We're going to learn about the cranial nerves that come off of the brain. Replace this with a thorough discussion of the structures of the eye and the physiology of vision. Next lab, you get to dissect an eye, so that's fun. So we're trying to prepare you for that in full force. 0 to 60 in 3 seconds. They do still do that, right? Okay, good. So, these are the things we're covering. Let's start with nervous tissue. Some of you will already know what a nerve is and what to look for. There are two main types of cells that we think of in the nervous system: neurons and neuroglia. Nerves and then support cells is essentially what those are. Neuroglia far outnumber neurons, and I'll show you what those look like on the coming slides. But the characteristic cell in the nervous system is the neuron. That's what we think of when we think of a nervous cell. The neurons largely all follow the same structure and breakdown. They have a cell body that contains the nucleus and other structures, dendrites that are sort of stubby projections, look like little cones, and they're highly branched. Those are inputs to the nerve. Dendrites are where signals arrive at a given nerve. And then the axon is a long, maybe cylindrical, what's the word I'm looking for? Progression? No. Projection is what I'm looking for. Where the signal leaves the nerve body and is transmitted to something else. Another nerve, perhaps, or muscle cell. So the dendrites receive, axon sends signals. Neuroglia support, and I'll talk about that just in a second. But you can see here what I mean when I talk about a neuron. The shaded beast is a neuron. The central region, which is sort of circular, but where the mass of the neuron lives, is the body. And we can see the projections coming off of that body and the circumference around it. So to the lower left, you might think, okay, these are dendrites. They are stubbier, a bit cone-shaped. They're input. Probably right. The axon, a little harder to decipher, and in a lot of cases you can't tell. So coming off of the left and the right are two projections that both look long, thin, somewhat cylindrical. And if you can't obviously tell the difference, we won't ask you to differentiate them on a test. Only if the differences are obvious, dendrites versus axon, will you be required to differentiate between the two. A lot of the time they're hard to differentiate, as is shown here. Not obvious if it's this one on the right or this one on the left. Probably not this one. The neuroglia are these little cell bodies around the neuron. And they're typically responsible for secreting fuel and nutrients. Myelin is part of the structure of nerves that we'll talk about shortly. It comes from neuroglia, and they far outnumber neurons. There's tens of billions of neuroglia I'm not sure for how many nerves, but there's many more. There's 85 billion nerves in the brain, many more neuroglia than that. Now what makes nerves special is that they're excitable. Nerves and muscle cells are excitable. We're going to run out of time with this cool new AV stuff. Nerves and muscle cells are excitable. And what we mean by excitable is they generate or respond to an electrical signal. Excitable. That electrical signal we've already introduced as an action potential. As an action potential. That is the name of the electrical signal itself. Now, it's not only important that they respond to that signal, but they can transmit that signal. Action potentials aren't static. They move through the neuron or through a muscle cell. That signal is propagated, which is equally important to just being responsive to an electrical signal. So they travel along the membrane of these cells, and then they interact with what we're going to call here voltage-gated channels. And all that means is that a channel will react, usually open, when the voltage of its surroundings reaches some threshold value. Thresholds are different, actions are different, the specific channels are different. Are they for different ions? That's not important yet, but you will know those intimately soon. So, at the risk of going long, I'm leaving this here. We are going to pick up on Thursday with our overview of the nervous system, branches of the nervous system,